Hello everyone, hope you all are doing great. This video is part 2 of the CFTRI previous year questions of 2019 and 2020. If you haven't watched the part 1 of this video, please go and check it out. This video is going to be helpful for all the CFTRI aspirants as it will help you practice the previous year questions as well as revise the important topics that have been asked in these questions. Also, even if you're not preparing for CFTRI and preparing for some other exam, these questions will give you a great idea about the important topics that you need to cover. So without further ado, let us begin. Before starting the first question, I want you all to take these questions as a quiz and try to solve it along with us. Once we discuss the question and the underlying concept related to this topic, I want you to pause the video and think for a moment and try to answer it yourself. This will not only boost your confidence but will also help you recall the topics and the concept that you have studied. Also, in the end, we have few practice questions for you, so stick till the end. So starting with the first question, we have non-protein organic molecule which bind at active site for enzyme catalysis is called. Now we have to see the keywords in this question. Here the question is asking about some molecules which are non-protein in nature and organic in nature. So there are some non-protein organic molecules which come and bind at the active site of the enzyme, and that is carrying out the enzyme catalysis, and that the molecule is called dash. We have to tell the name of the molecule. Now you can decode that this question is from the enzymes topic, and I'm sure you must be knowing what enzymes are. They are the biocatalysts which accelerate the chemical reactions or the biochemical reactions. Now these enzymes are very specific in nature and they have a very special site called active site, which you can imagine it like a cleft or some kind of opening where the substrate is actually going to come and bind and where the reaction is going to proceed. Now not all the enzymes are capable of carrying out the reaction on their own. They require some helper molecules. Now these helper molecules are called cofactors. So what are cofactors? Cofactors are basically non-protein chemical compounds which are helping the enzymes to carry out the reaction. So they are non-protein in nature. This is very important point that you have to know. You must be knowing that enzymes, mostly the enzymes are protein in nature. Some of them are RNA based, but mostly enzymes are protein in nature. While these cofactors or these helper molecules are non-protein in nature, right? Now there are two terms, very important terms when it comes to the cofactor. There are two terms related to it. When the enzyme or the cofactor are attached to each other, that is when the cofactor has come and bound with the enzyme, this whole set is called holoenzyme. And if the cofactor has not been attached to this enzyme, it is simply called the apoenzyme. There have been a lot of questions which have been asked about what holoenzymes are and what apoenzymes are. It might get a little confusing, so you have to remember that holoenzymes is something when the cofactor is attached to the enzyme, that is the non-protein molecule or the helper molecule has been attached to the enzyme, and when it is not attached, it is simply apoenzyme. Now these cofactors can be of two types. First is they can be inorganic in nature, like some metal ions or like zinc, magnesium, iron, copper, etc. And they can be organic in nature. Organic means they are having carbon atom in them. In that case, when they are organic, they are called as coenzymes. Examples include your NAD or NADP, that is nicotinamide, adenine dinucleotide, and other coenzymes are there. Now here only, if you pause, if you see, we have got the answer. What we are talking about, the question was asking about some non-protein molecules which are organic in nature and they are binding at the active site of this enzyme. So this is what we have, the organic in nature molecules, and they are non-protein as well. So coenzyme is actually a type of cofactor only, but you will not mention cofactor as the answer over here. The answer will be coenzyme because the question is specifically talking about the organic molecules. Let us see a little more about this. Coenzymes bind loosely to the active site by some reactions and by some bondings, and they help to catalyze the reaction. If you see in this image, once the enzyme is there, this is the apoenzyme. Once the cofactor or coenzyme binds at the active site, it will get activated and it will form the holoenzyme. And after activation, now the substrate can come and bind, and now the reaction can proceed further. So this is how the reaction of this enzyme will occur. So here we have the answer as coenzymes. Now, talking about this particular topic, when it comes to enzyme, like I told you, this enzyme and this cofactor concept is very important. In another year in CFTRI only, there was a question talking about what cofactors are, whether they are protein molecules, non-protein molecules, or sulfur-containing molecules. Now, I'm sure you must be able to answer that cofactors are non-protein molecules. Also, in enzymes, there are other areas which are very important, such as the application of the enzymes. Industrial applications of the enzyme have been asked many times in many of the exams. exams including CFTRI exam. For example, questions such as which enzyme gets inactivated during pasteurization of the milk, or which enzyme is the indicator of the complete pasteurization of the milk. So you can answer those questions also. So you have to make a list of all the enzymes which are used in the industry, for what purpose they are used, and they can be asked in multiple ways in that way. Now we'll move on to the next question. The next question is, which of the following will not help in prevention of pellagra? So if you have heard about this term, you must be knowing that it is a deficiency disorder. If you haven't, don't worry, we'll talk about it in detail. So now they are asking that what chemicals or which of the compounds will not help in the prevention of the pellagra. So you have to be very careful while reading the question. The question is asking about a negative answer. So which of these will not help in prevention? To answer this, we should know what will help in prevention of pellagra, and to get that also, we should know what pellagra is and how it is caused, what deficiency disorder it is, right? So let's discuss about pellagra before answering the question. So pellagra is basically a systemic disorder or a disease caused by a severe deficiency of niacin. Systemic disease means it affects the whole body, and this is caused by deficiency of which vitamin? Vitamin B3, that is niacin. So vitamin B3 is called niacin, and if there is a deficiency of niacin in your body, it may cause pellagra, right? Now the symptoms of pellagra, it affects the whole body. If you see there, it affects the skin, means a very peculiar type of symptom of pellagra is that there's redness over the whole neck, and that is called Cassell's neck disease. So that happens, it affects the mouth, it affects the bowels, there will be diarrhea in case of pellagra, and also it affects the brain. There are cases of dementia and depression and others related to this pellagra. Now, talking about niacin, we know that now pellagra is caused by deficiency of niacin. Now, niacin is basically a generic term for the two compounds, nicotinic acid and nicotinamide. So, there are these two chemical compounds. They are both vitamin B3 forms only. Nicotinamide is also called as niacinamide. So, if whether you see nicotinic acid or nicotinamide, these both are vitamin 3 forms only. Now, this niacin is very important vitamin. Why? Because it is involved in metabolism of macronutrients such as your carbohydrates or your proteins or fats. Now, why it is helping? So, because it is forming a part in a very important coenzyme which is NAD and NADP. Now, we already know what coenzymes are. We have already seen in the previous slide what coenzymes are. They are helping the enzyme to carry out the reaction. So, this vitamin is forming some coenzymes. These coenzymes are very much important for the metabolic reactions in our body. So, if you see the chain, if this vitamin is not there, these coenzymes will not be in sufficient amount and your metabolism will get affected. That is how the whole body gets affected if there is a vitamin deficiency or there is a vitamin B3 deficiency. Okay, now sources of niacin include fish, meat, cereals and legumes. Basically, all the proteinaceous foods are great source of niacin. And also this niacin, the good thing about that, it can be produced in our body as well. How? This niacin can be produced from essential amino acid tryptophan in our liver. So tryptophan can get converted through series of reaction into niacin. How much tryptophan gets converted into niacin? 60 mg of tryptophan can make up to 1 mg of niacin. So now, by all this discussion, we have understood one thing, that for this prevention of pellagra, what we would need? We would need a food which is rich in niacin and tryptophan that will help prevent the pellagra disease. Now, let's try to look at the options. We have niacin over here. Nicotine, we don't know. Try to be very careful while reading the terms. We are not saying nicotinic acid, it is nicotine. Now, this nicotine is actually present in your tobacco products. If you have heard about it, it is a chemical compound present in the tobacco products. Nicotinamide is actually a form of niacin. So, yes, it will help prevent your pellagra disease, and tryptophan again can help prevent it. How? Because it will get converted into niacin, and niacin will help prevent pellagra. Okay, so here we have got the answer that which will not help in prevention. The nicotine is not going to help in prevention because that is not related to this niacin. Okay, so this is how you're going to see the related concepts of this particular question. Now, talking about this vitamins, all the vitamins and the nutritional aspect of it, the deficiency disorders aspect of it become very, very important for all kind of examination, and there are multiple questions on various type of deficiency disorders. If you see the next question, again it is related to a deficiency of a very important vitamin, that is folic acid. So the question is saying that deficiency of folic acid causes which of the following disease? Night blindness, megaloblastic anemia, beriberi or rickets. Now let's try to see again about this folic acid, and we'll see what it is causing and where it is, what role it is playing, and that is how we're going to derive that how this deficiency disease happens. So folic acid is basically vitamin B9, which is required for a lot of function. It has multiple functions such as making RBC. It is helping in making the red blood cells. It is helping in the cell division and the growth, and also helping in RNA and DNA synthesis, which will affect and ultimately also going to affect the brain activities. So if you see, they are involved in multiple roles, so their deficiency is also going to be multiple. So if you see, one important deficiency of folic acid is basically it can cause megaloblastic anemia or macrocytic anemia. So the name itself is telling you it's some sort of anemia. How anemia happens? When there is low red blood cells in the body. How low red blood cells will happen? Because we saw that this vitamin B9 is actually required for making the RBC. So if there is low folic acid, less RBCs or less mature RBCs will be produced, and this less mature RBCs are quite large in size. So that is why we call it megaloblastic, because RBCs will be very large in size, or we can say macrocytic. Macrocytic means the cell size is very large. So macrocytic anemia or megaloblastic anemia is happening due to deficiency of folic acid. Okay, so folic acid deficiency can cause other effects also, such as in pregnant women especially, where there is development of the fetus is required. If folic acid is deficient, then it can cause a lot of fetal neural tube defects, such as spina bifida is one such example. So because it is involved in division and growth, it can affect the neural growth, or fetal growth as well. Okay, so here we have got the answer out of these all four options. Which will be the correct answer? Yes, the answer would be megaloblastic anemia, because it will cause the immature red blood cells, which are very large in size. We'll see about the other deficiency disorders also. Do not worry if you do not know about these. Like I said, it is very important to know about all the deficiency disorders, because multiple times questions are asked from one or the other vitamin. So what we have done is we have brought all of these vitamins all together and indicating their deficiency disease. You should remember all the deficiency disease, and also about one very important or something very peculiar symptom of that disease. Okay, so talking about vitamin A, it can cause night blindness. The deficiency of vitamin A can cause night blindness or xerophthalmia. Xerophthalmia is basically keratinization or pigmentation in the cornea. It becomes very pigmented. So xerophthalmia has been asked in CFTRI in one of the year. It was asked that xerophthalmia is caused by deficiency of which of the vitamins. So now you know that it is caused by vitamin A deficiency. Deficiency of vitamin A can also called keratomalacia, means there is cloudiness in the cornea. So that is one disease. Vitamin B1, that is thiamine, the deficiency of it can cause beriberi. Beriberi actually literally means I can't. So your body is saying I can't. So it affects the whole body again. Vitamin B family is actually impacting a lot of body parts. So the body becomes very weak, and there are multiple disorders related to it. Vitamin B2 is riboflavin. Deficiency of riboflavin can cause a riboflavinosis, means it affects basically the mouth area. There are sores in the mouth. It can cause glossitis, that is tongue inflammation, cheilosis, that is cracking of the sides of the mouth, and stomatitis, that is mouth inflammation. Vitamin B3, we have just now discussed that niacin can cause deficiency disorder, the pellagra. Vitamin B5, that is pantothenic acid, the deficiency of B5 can cause burning feet. Vitamin B6 deficiency can cause hypochromic microcytic anemia. Means, if you break down the terms, hypochromic, hypo means less, chromic means color. So this anemia, again we are talking about RBCs only. Wherever this anemia comes, you have to think about RBCs. So this hypochromic means the red blood cells have less of color, and they are micro, means very small in size. In folic acid, we saw that it is megaloblastic or macrocytic. Macro means they are very large in size, while in pyridoxine or in vitamin B6, it is very small in size. So RBCs with small size and red color is a symptom of deficiency of vitamin B6. While vitamin B7, that is biotin, it affects if deficiency of this can cause dermatitis, that is the redness or itchiness in the skin. In case of folic acid, that is B9, we have discussed, it can cause the megaloblastic anemia and some of the neural tube defects as well. In vitamin B12, cyanocobalamin, the deficiency of B12 can cause pernicious anemia. Now, what is pernicious anemia? Pernicious anemia means there is again low red blood cells in the body because of poor absorption of vitamin B12. Again, it can also cause megaloblastic anemia. Again, vitamin B12 become very important for pregnant women as well. Okay, next is the vitamin C, that is ascorbic acid. Deficiency of it can cause scurvy. We all know about it, that bleeding gums and weak muscles, all of this are symptoms of scurvy. Vitamin D, that is calciferol. Vitamin D can cause deficiency disorders such as rickets. In children and in adults, it is called osteomalacia. Vitamin E, that is tocopherols, it can cause muscular dystrophy, hemolytic anemia. Hemolytic anemia means faster depletion of red blood cells takes place in the body. Vitamin K, again we know that vitamin K is involved in the clotting of the blood. So if there is a low vitamin K in the body, it can cause bleeding, diathesis, or hemorrhage can also occur. So these are all the deficiency disorders. You can take a screenshot and keep it with you and try to revise before your exams, right? Moving on to the next question, the amino acid used for synthesis of niacin. We have discussed this question a few minutes back only. Now you can answer this if you pause the video, you can answer this question very easily, that niacin, how niacin can be produced from an amino acid. So we have discussed that niacin exists in two forms, that is nicotinic acid and nicotinamide, and it can be formed in body and liver from amino acid tryptophan. It's an essential amino acid. So if you consume this tryptophan, it is required for the niacin production as well in the body. So for this conversion from tryptophan to niacin, other mineral or other vitamins are required, such as riboflavin and vitamin B6 are required in these reactions. And how much amount of tryptophan will make how much amount of niacin? We have seen that 60 mg of tryptophan is going to produce 1 mg of niacin. Also, to account for this amount of niacin or tryptophan in foods, a particular term, that is niacin equivalent, is used. So one niacin equivalent is actually 1 mg of niacin and simply 60 mg of tryptophan. So now you can answer this, that which of the following is the amino acid which is producing the niacin? It will be tryptophan will be the correct answer over here. Okay, let's move on to the next question. This question is talking about the RDA of folic acid for pregnant women. Again, in nutrition, we saw one aspect, that is about the vitamins, the deficiency disorders, those things are there. The other thing is there is the RDA. The RDA, that is the recommended dietary allowance. It becomes very important concept. Now, ICMR recently in 2020 revised the RDA. For all the nutrients, there are some revisions, so you need to refer to that latest revised RDA and you have to answer these kind of questions. Of course, you cannot memorize all the values of all the RDA, but for the important nutrients and especially for the important categories, it is very important to remember the RDA. So important categories as in where there is increased amount of nutrition or that particular nutrient is required. So in that case, it becomes very important. So here you can see pregnant women, we know folic acid is required. We just saw that folic acid, if there is deficiency of it, it can cause neural tube defects, neural fetal tube defects. So to prevent that, folic acid is required in large quantities by pregnant ladies. So what is the RDA? The question has asked about it. Let us see as per the latest data of ICMR 2020. So the RDA for the pregnant woman is 570 microgram per day. So that will be our correct answer over here. Okay. Moving on to the next question, recommended dietary fiber intake per day is, now again they are asking for the how much dietary fiber should be taken in a day. So again there are different values for the fiber as per the different age groups. What ICMR has told us, that about 30 grams per 2000 kilocalorie is considered to be safe for the fiber content, and generally there is a consensus understanding that 25 to 30 grams per day is required for the fiber. Now if you consume too much of fiber, or let us say even if for other nutrients also, too much of any nutrient can cause other related problems. If you consume too much of fiber, it can cause bloating, gas, cramping, and if there is deficiency of fiber, it can cause constipation, irritable bowel syndrome, and even some cases of cancers can happen in deficiency of this fiber. So here we have got the right answer. As per ICMR, it is 30 and general range is also 25 to 30. So you can see what 30 lies in this range. So option B becomes our correct answer. Okay. Moving on to the next question. The next question says microorganisms growing in high pressure are which of these? Osmophiles, pyrogenic, piezophils, halophiles. If you do not know these terms, don't worry about it. We'll talk about them in detail. First, talking about the microorganisms. We know that microorganisms require some ambient conditions for their optimum growth. Let us say the temperature, some pH, some water activity, and some pressure conditions for their growth. But some varieties of microorganisms can even grow in extreme conditions. We know them. They are called extremophiles. They can grow in extreme temperatures, extreme pressures. Now this question is talking about such a microorganism only, which is growing in very high pressure. So we have to tell the name of that particular microorganism, what they are generally called. So there are different terms for these extremophile. For in case of temperature, they have different terms. In case of pressure, water activity, they have different terms. So to answer this question, we'll first understand the concept in detail. So this graph with this flowchart will help you understand what different microorganisms are called when they are living in extreme conditions. Okay, so in case of extreme temperatures, we know that very low the microorganisms which are growing in very low temperatures, they are called psychrophiles, means low temperature loving microorganisms. They can grow below 15 degrees Celsius, can even go to 0 or even less temperatures. The next we have psychrotrophs. Now there is a lot of confusion among students about psychrophiles and psychrotrophs. See, psychrotrophs are those microorganisms which have optimum growth in the normal temperature range only. That is, in the mesophilic range they can grow well, but they can also grow at lower temperatures, means they can grow if the temperatures are decreased, they can grow about 0 to 7 degrees Celsius as well. So that is why they are also called as facultative psychrophiles, means in some conditions they can adapt to that condition and they can grow as psychrophiles. Okay, or they are sometimes also called psychrotolerants also, that they can tolerate the low temperatures. Next is the mesophiles. They are growing at the normal temperature range about 20 to 45 degrees Celsius. Thermophiles at higher temperatures, and hypothermophiles are growing at extreme higher temperatures at about more than 80 degrees Celsius, can even go to 100 degrees Celsius or even more than that, right? Now in case of extreme pH, what will be the extreme pH? The normal pH range is the neutral range where the microorganisms can grow well, but even in extreme acidic conditions and extreme basic conditions, the microorganisms can grow, some of them, not all of course. So those microorganisms which can grow at very low temperatures are called acidophiles. They can grow in acidic conditions such as pH below 2. Alkali philes will be those which are growing at high pH conditions or in alkaline conditions in about pH 9 to 11. Next, talking about the water activity, we know that microorganisms can grow well in high water activity. So the foods which are liquid, they have high free water, the free water is available in them, so the microorganisms can grow well in them. But as the water activity reduces, it becomes difficult for the microorganism to grow. Again, we have some special microorganisms which can adapt and which can grow in low water activity as well. So they are called xerophiles, which can grow in water activity below 0.8. Okay, now talking about solute concentration, very high solute concentrations when are there, such as in case of very high salt concentration is there, those organisms which can grow in such conditions are halophiles. A very good example would be those microorganisms living in the ocean. See how much amount of salt is there in the ocean and they can still survive in those high salt conditions. Okay, the next is the osmophiles, means they can sustain very high osmotic pressure like sugar concentrations. So they can grow well in high sugar concentrations or high osmotic pressures, and they are called osmophiles. They love such kind of conditions and they grow optimally in such conditions. Next, we come at the extreme pressure conditions. So the microorganisms which can grow in extreme pressure conditions are called piezophiles or the barophiles. See, piezo term over here is coming from the term squeeze or very high pressure kind of word. So means that piezophiles, they can sustain very high pressure. Our barophiles, bar we know that is the unit of pressure, so we can also call them barophiles. They love such kind of condition. They can grow well in such kind of high pressure conditions. So after knowing all of these extreme conditions, now I'm sure you'll be able to answer any question related to these microorganisms. So let us get back to our question, what it was, which was asked in CFTRI. So microorganisms growing in high pressure. Now you'll be able to answer this question very easily. So they are called, what they are called? They are called piezophiles. Let us talk a little about piezophiles, that they can sustain high hydrostatic pressure and they are very commonly found in the deep down oceans because the pressure in oceans is very, very high. Hydrostatic pressure is there. And piezophiles, of course, as we discussed, that they can sustain high pressures above atmospheric pressures. There are some microorganisms which can grow at both normal temperature, normal pressure conditions as well as high pressure conditions. So those will be called piezo-tolerant microorganisms. Okay, moving on to the next question. It is related to microbiology. The question is asking about generation time is calculated by which of the following formula. Now for such question, you need to know the formula for this particular concept, for this particular generation time. Before getting to the formula, we'll talk about what generation time is, because again it is very important topic in the microbiology. So generation time is basically the time required for the cells to divide, or we can say in how much time the whole population is doubling, because we know that bacteria or the microorganisms are basically dividing by a binary fission. So they are doubling actually 1 to 2, 2 to 4 and further on. So in how much time this whole population is doubling, that time taken for this new generation to be formed, that is called generation time, or the time required for the cells to divide. Now generation, we know that they are growing by binary fission, as we said, 1 to 2 you can see, then they are 2 to 4 they are going, 4 to 8. So this, they are growing in geometric progression, or in exponential terms if you write this, we can say 2:0 or 2:1. Again in multiples of 2 only they are growing, where n at the end we'll have 2 raised to the power n will be the total population, and n will be the number of generation, in which generation this population we can get to know. Such as here, if the 4th generation, I ask you how much, if the initial population was 1, in the 4th generation what would be the total population. So we can simply write 2 raised to the power 4, you will get the answer as 16. Now, talking about generation time formula, there is a very simple formula for the generation time which talks about the total time for this initial population to convert into the final population. So, time divided by 3.3 log small b by capital B. Small b here is the final population. So, that is at the end of the interval T, what is the number of bacteria or the population cell, how many cells are there, and capital B is actually the bacterial population at the beginning. Now, for those of you who can remember this formula, well and good. For those of you who want to know actually how this formula has been derived, so we'll just quickly see how this formula has been derived to better understand this generation time formula. So here only we have got the answer that first option is the correct answer over here, because this is the formula for the generation time. Okay. Let us see this formula. How generation time, we said that how much time is taking for this cells to divide, the population to double. So T upon N, that is how number of generations are being produced in how much time, that will give you the generation time. Initial population we have capital V, small b is the final population, and N will be the number of generations that have been produced from the initial population, right? Now to get the final population, this general formula you can derive very easily, because the final population is going to be some initial population into some divisions that have happened. That is 2 raised to the power N is the multiplication factor, that how the population is growing, how the generations are being growing. So that will give you the final population. Now this we are going to solve this to find the value of N actually. So if you take the log on both the sides, you will get this equation, log small b is equals to log capital B plus, because the power will come n into log 2. Now if you further solve this, you will get this equation that N value is equals to 3.3 log B upon capital B. Now if you put this value of N, that is the number of generation, in the G value, that is the generation time, you will simply get this value of the generation time. So this is the formula for the generation time. Now we move to the next question, that is related to the processing. HPP, a non-thermal processing, stands for, so what is HPP? They are asking that what it stands for. High performance processing, high pressure processing, heavy product processing, hot product processing. It's very simple question. You must be knowing what HPP stands for, and it's a non-thermal process. HPP stands for high pressure processing. Let's see what HPP is. So HPP, as the name is indicating, it's a kind of processing where very high pressures are being used. So it is a non-thermal processing. Why? Because no heat treatments are being given, only very high temperatures are used to inactivate the microorganisms. It also yields similar effects such as pasteurization, because again the microorganisms are getting killed. So, but no heat is being involved. So, it is also called cold pasteurization, because pasteurization is happening without any heat treatment. Okay, it is a non-thermal process with the help of some very high pressure. How much high pressure is there? About 6,000 bar, up to that high pressures can be used. Also, because no heat is being used, the food nutritional quality and sensory quality is not affected at all. Okay, so the correct option over here would be option B, that is high pressure processing. Very simple question. We'll move on to the next question. Conduction is used as heat transfer in which of the following foods? Again, very simple question. It is asking about in which of the foods conduction will be the heat transfer mechanism. In which of the foods conduction will be the main source of transferring heat. So these foods include meat, fruit juice, emulsion, none. You have to answer out of these. Now again, very simple. You must be knowing about the heat transfer mechanisms. It can happen through conduction or convection or radiation. So I need not tell you what conduction, convection, radiations are, but you must be knowing that conduction happens when there is direct contact between the objects, and convection happens in case of fluid, where there is a movement of the fluid, and in case of radiations, electromagnetic radiations are being used to heat the food. So now conduction, if you see, it involves the transfer of heat at the molecular level, and actually there is no physical movement of the object, and this will happen when there is direct contact, the objects are in direct contact with each other. Now among these food, you can easily say that where the food will be in direct contact with each other and where the food is not moving actually, and it happens basically in solid foods. The conduction we know it happens in case of solid foods, while convection, as we said, it happens in case of fluids. So here it's very easy which of this is solid. Meat is the solid one, because fruit juice and emulsion are liquid, we know that. So here the correct option will be meat. Very simple question. Again, let's move to the next question. dash food contains highest protein content. So options are mushroom, meat, milk, wheat. Which of the foods contain highest protein content? You have to answer that. For that, you need to have an idea about how much protein content is there in each food. Although again, it's very simple. You can guess also which of the foods is highly proteinaceous food. But let us see in detail how much proteins are there in each food. If you see in mushrooms, we have about 2 to 3%. In milk, 3 to 4%. In wheat, again, depending upon the variety of the wheat, if it is soft wheat, it will be 9%. If it is hard wheat, it is about 13 to 14%. In meat, again, depending upon the type of the meat or which animal the meat has been procured, the protein content varies. On an average, we can say about 23% protein is there in the meat. So from this only, I've got an idea that highest protein content is there in the meat. Also talking about meat proteins, we know that animal proteins are very good source of protein. Why? Because they are called complete proteins. Why? Because they have full set of essential amino acids. All the essential amino acids are there in those meat proteins, animal proteins, such as meat, eggs, milk, etc. So they provide very high quality of protein sources. Okay, now in this question, the option will be meat because that is having the highest protein content. Okay, moving on to the next question. It is talking about partially fermented tea. So partially fermented tea is which of the following? Oolong tea, green tea, black tea, yellow tea. For these type of questions, again, the plantation crops and other processing procedure or the steps of the major crops and products, it is very important for you to understand the different varieties and different processing steps which are there. So let us talk about these different type of tea. We know that different type of tea are there. The basic plant remains the same. The leaves are plucked. That leaves till there is the same. How the different tea are being produced? Because the different processing steps that are being further used. So depending upon this processing, we have different teas. There are a lot of different type of tea, but we'll discuss these 4 major tea. The most common ones, basically the first one is the black tea, which is commonly consumed in India. So black tea is produced by fermentation or oxidation process. If you see the process of the black tea, first the leaves are plucked, they are withered, and then they are crushed. And how this crushing, after that they left exposed. Why they left exposed? So that enzymes can get activated and they can act on the juices of these leaves, and that will cause the oxidation process. So there the color will develop, aroma will develop, and that is how the tea leaves turn into dark leaves. After that they will be dried and they will be packed and sold. So there this oxidation step in like in general processing units, this step is called fermentation, though technically it is not the fermentation as you think of microorganisms. It is technically actually the oxidation process that is happening because of the enzymes. So that oxidation or fermentation takes place in that tea, and that develops the color of the tea. So next tea, talking about the next tea, that is green tea. Green tea is totally unfermented tea. This whole step of oxidation or the fermentation has been omitted in this step, in this processing. What happens once the tea leaves are plucked, they are steamed or they are heated so as to inactivate the enzyme. So we do not want the enzymes to come and to get activated and act on these leaves and turn them into black. We want these leaves to stay green only. So that step is completely omitted, and after that they are simply dried and rolled and dried and then sold. Next tea is the oolong tea. Oolong tea, you can think in between these black and green tea. It is semi-fermented, not the complete oxidation by the enzymes takes place, and not even it is not there. There is no oxidation. There is oxidation to some extent. This fermentation step is too short for changing the color to completely black. So that tea is called oolong tea. The next tea is yellow tea. Again, yellow tea is quite similar to these tea, the oolong tea, but it is very lightly fermented. You can say it is more like of a green tea, but a little fermentation is over there. In this step, the special type of procedures are used, such as the enzymes are inactivated first, like in case of green tea, we are inactivating by firing and heating, and after that there is sweltering. Sweltering means that the leaves are wrapped and piled on. That is called wet piling that happens, and during that a little color development takes place. So during this tea, the tea color changes. During this process, the tea color changes to a slight yellow, and that is how the name has come, yellow tea. So out of these, which one is partially fermented or semi-fermentation? Proper fermentation step has been done, but partial, not full fermentation has been done. So which will be the tea? We have got to know from here only that oolong tea is the right answer. Oolong tea is the tea where there is partial or semi-fermented tea it is. Okay, so the option A is the correct answer. Moving on to the next question, it is about mother's milk secreted on 1-2 days is called what? Casein, whey, water, colostrum. Again, very simple question. If you look at the options given, these casein, whey and water, these are the components of the milk, not the type of milk. They're talking about the mother's milk which is secreted initially after giving birth on 1 or 2 days, on the first or second day. That milk is called colostrum milk. Let us see what colostrum milk is. So as we said, it is released by mammary glands of the mother after giving giving birth on the first or second day. Why this colostrum milk is so unique and why it becomes important? Because it is rich in nutrients and antioxidants and antibodies, so that it gives the newborn baby the immune to fight the diseases in the initial days. Also, compared to the regular milk, this colostrum is very thicker and very yellow in color. So the correct answer over here, it's very simple, will be the colostrum milk is the correct answer, option D. Moving on to the next question, delayed complementary food to infant causes, what it will cause? Now again, simply without seeing the options, you can answer this, that if you not give the complementary food on time to the infants, to the babies, what will happen? Of course, the growth will get affected. So here, let's talk about complementary food over here. Complementary food is basically needed when the baby is growing, when the breast milk or the infant formula, which the baby has been receiving for the initial 6 months, is no longer sufficient for meeting the growing requirements or the nutritional and development requirements. So the complementary food has to be introduced to around at the age of 6 months, and if delayed introduction of this complementary food happens, it can affect the baby's growth definitely. So the correct option over here of this question will be low growth will happen if complementary food is delayed. Moving on to the next question, very easy question and the last question of this PPT. The highest citric acid content is in citrus, gooseberry, mango, berries. Very simple question, even you can tell it without even seeing the options, that highest citric acid is found in citrus fruits. Even the citric acid word has come from the citrus fruits only. So we know that citric acid content is very high in citrus fruits such as lime, lemon, orange, tangerine, etc. Now we have few practice questions for you. Try to solve them and give the answers in the comment section below. I hope this video was helpful for you. If you found it helpful, please like the video and share it among other aspirants also. Thank you so much for watching the video.